Let's look at an example of transcriptional control in a prokaryotic organism. In this example, we will examine the trip operon. This operon is referred to as a repressible operon because it can be turned off or repressed when the levels of tryptophan are sufficient for the bacterial cell. Before we look at the specific trip operon, we need to explain what an operon is. It turns out that bacterial genes are organized somewhat differently than eukaryotic genes. In bacteria, the operons are locations on a chromosome where the genes for a certain function are found physically together along with their controlling elements. In the case of the trip operon, 5 genes that encode for 3 holoenzymes required for the synthesis of tryptophan and the operator sequence to control them are closely linked and known as the trip operon. When the trip operon is turned on, it allows the production of the enzymes that will manufacture tryptophan, an amino acid. So, on a part of the bacterial chromosome, there is a stretch with genes 1, 2, 3, 4, and 5. They are all involved in the manufacture of tryptophan. Just upstream of these is the operator sequence, shown as start. And upstream of that, outside the operon, is the trip repressor gene, which is always turned on, but the gene product, the protein, is inactive. Under low tryptophan conditions, RNA polymerase binds to the operator, labeled start, and transcribes each gene in sequence 1, 2, 3, 4, and 5. And they are translated into their respective polypeptides. These will form the enzymes necessary to build the amino acid tryptophan. Eventually, a tryptophan will bind to the repressor protein, and this activates the repressor. With tryptophan bound to it, the active repressor binds to the operator site and blocks the RNA polymerase from transcribing the trip genes. And so, the trip operon is an example of a repressible regulation. When tryptophan is present, it turns off the operon that would manufacture tryptophan. When tryptophan levels are low, then the repression stops and the enzymes to build tryptophan are produced. It's important to remember that the trip repressor is always produced, but only becomes active when bound to tryptophan.